So since microbiology is a biohazardous lab, no food or drinks were allowed to be consumed in the lab, right, because it's hazardous. And the bacteria that we use are not really pathogenic. And what does pathogenic mean? What's a pathogen? Right, it can cause disease. So, right, Rachel. So, we're not really working with those kinds of microorganisms. So, there really wasn't much of a risk, but we still wanted you to treat those microorganisms as if they were pathogens, as if they were dangerous. So that way you would know how to do things and how to do things safely. So you never ingest, smell, or mouth pipette bacterial cultures because there is a risk when you do that. And if you were to spill anything or break anything, you had to let me know because if it involves live bacteria, there were procedures we had to follow to clean it up. No horseplay in the lab, that means no fooling around, no scaring your lab partner, things like that. And if you had long hair, you had to keep it tied back. And that's because we do use Bunsen burners a lot. So no hair around fire, and also hair poses a contamination risk. So we would have asked you to tie your hair back. Shoes, I make a big deal about shoes because you are in a hazardous lab setting. You should always wear shoes that cover the entire foot. Slippers and ballerinas, flats, those shoes did not cover the top of your foot, so those were not acceptable. So you needed a shoe that covered everything, including the top of your foot, so like a sneaker. That would have been fine. No sandals, flip-flops, nothing like that. Like I said, your entire foot had to be covered. You guys luckily don't have to worry about that because you're not in lab. But in the future, in other lab courses you take, there will be similar requirements. Eye protection. You were required to wear goggles in the micro lab because whenever you're using fire, live bacteria, or toxic chemicals, you had to wear goggles to protect your eyes. You also had to wear aprons. Now, we are not doing the handout on where to put things, but normally we would have included a handout, where do I put it, because you needed to know where everything went in the micro lab. Since we're not actually meeting in lab, we're not going to bother doing that, but everything had a specific place where it had to go. You had to know about biohazard bags, but we're not actually doing that remotely, so we're not going to bother. And when it came to the microscopes, your seats had a number on them, and that corresponded to the microscope number that you would use. But we're not using microscopes, so. And then, of course, the next page is something that you would have filled out and signed and given to me. I'm not going to do that this semester, but that's what you would have done. I may ask you later just to tell me about yourself, but for now, we're not going to bother with that. But lab safety rules and regulations are important, so you should know about them, because even if we're not in lab this semester, at some point in the future, you will be in other types of labs, so you do have to follow proper procedures. And normally what I would do is, if you're doing something wrong, I would correct you. So I would constantly get on your case to do things right. And it's not because I was picking on you, it's because it matters. Knowing how to do things correctly and safely matters. Because when you leave this course, you're going to be in a situation where you have to follow proper procedures. And if you don't follow proper procedures, what's going to happen? So tell me, what's going to happen if you don't follow proper procedures? Wherever you are, in a course, in your job, what's going to happen? So tell me, if you don't do things right. Yeah, what kind of problems, Maria? Yeah, what may go wrong, Christiana? Right, exactly, Carice. So, yeah, and Taylor, who get fired too. Right. So, yes, you may get fired, but if you're a nurse or a nursing assistant or something else, and you don't follow proper procedures, you may injure or kill a patient, or you may infect yourself with something. So, paying attention to what you do in lab matters because when you leave here, it will matter. So learning how to always think about what you're doing in lab is what I try to emphasize in the micro lab. Because when you leave here, it will matter. So it's best to learn these lessons early, right? So you don't get fired or you don't injure someone or you don't infect yourself. So that's why lab safety rules are important. You need to follow proper procedures. When you don't, things happen. And of course, right now, if you didn't follow proper procedures, you could get infected with COVID-19, right? That's why we all should be wearing masks when we're out around other people. You're going to the grocery store, things like that. So, sound good? Any questions on lab safety? All right, so let's shut this one down. And then, so laboratory basics. So normally what we would do for lab basics is it would be given to you as homework, not something that was turned in and graded, but just something for you to work on. And the main idea behind the lab basics was just to familiarize you with the different types of equipment used by microbiologists and the different types of media that we use. So I'm just going to go through this handout with you now. We have nice colored pictures of things so you can see the kind of stuff that you would have played with if we were able to meet face to face. So how do we grow microorganisms? Well, a culture is the growth of bacteria or any microorganism for the purpose of study. And we use a culture medium to grow those microorganisms. And a culture medium is a mixture of nutrients. If it's liquid, it's a broth. If it's solid, it's agar. And that medium must be sterilized first. So your medium must be sterile so that only the bacteria or microorganisms that you introduce will grow there. So we always use sterilized media. So here you see a picture of a broth tube. So we use broth a lot. Here we have an agar slant and an agar deep. And so remember, the only difference between the broth and the agar is that we add agar to the broth to make it solid. So otherwise, all the nutrients are the same. It's just a solid surface on which bacteria can grow. Now, an agar slant and an agar deep use the same amount of agar, but before the agar cools, you can lay the tubes on their sides so they are slanted and it solidifies like this. Why would you want to use a slant? What's the benefit of a slant?
Is it more space for them for the bacteria to grow?
Right, so we call that space surface area, right? So we have a lot more surface area on a slant than we do a deep. Even though we're using the same amount of agar, we get all this surface area when we slant it. And so that makes growing a culture more efficient. You can grow it in a small, location, so you can store a bunch of tubes more easily. So that's why we use the agar slants. Deeps have their own special uses. Sometimes we will actually melt the agar so the students can make their own agar plates. But mostly we stab the deeps with bacteria and then observe a characteristic in that deep. So here we see broth tubes. Here we have an uninoculated tube, so the broth is crystal clear. Here we have bacteria growing, so the broth is very cloudy, and that tells you there's something growing in it. In this agar slant, there's nothing growing in the tube on the left, but there is a culture growing surface on the right. So you can see there's stuff, growing on the surface here, and there's nothing here, because you can actually kind of see through the agar here. Now, we do use Peachy plates. Here we have an empty sterile plate. Here's one with agar in it. The agar we use most commonly is TSA, and that stands for trypticase soy agar, which you don't really have to remember, but it's a good general all-purpose medium. Many kinds of microorganisms will grow on it, but not everything. It doesn't have enough nutrients to support the growth of every kind of microorganism, but it does support the growth of the most common ones, and of course the ones that we use. Here, in fact, you see bacteria growing on this agar plate. And we can see colonies here, individual colonies, and here the growth is really thick. But here we have the individual colonies. In this particular plate here, sometimes we pour other stuff on top of the agar, usually a melted softer agar that will solidify on top. Now, you see this label here, TSA? That's actually on the bottom of the plate. Here, we're actually looking through the bottom of the plate. So agar plates actually spend most of their lives upside down. So we often will inoculate them when they're upside down. We lift the bottom of the plate up out of the lid, and then we will inoculate the surface very quickly and turn it back over. We incubate plates upside down. And the reason why is that agar is kind of like Jell-O. It's mostly water. And the incubator temperature is 37 degrees Celsius. What's the significance of 37 degrees Celsius? What's 37? I know you guys know what 37 is. Nope. We'd be awful hot and sweaty if it were room temperature.
Normal body temperature.
It's body temperature, right? So what is 37 degrees in Fahrenheit?
98.6.
So 98.6, so that's pretty hot. It's almost 99 degrees. If you were sitting in your home right now and it's 99 degrees, would you be happy? It would be extremely unpleasant. It's very hot. So the problem with the agar plate, if you put the plate in right side up, then water will evaporate and condense on the lid. And when that happens, it will make a mess. So we don't want any condensation on the lid. So when you turn the plate upside down, there's a whole lot less condensation that can form on the lid. Also, as you can see from this plastic lid here, it just sits on top. It doesn't seal. So if you have a lot of condensation and you flip the plate over, it's all going to leak out all over and cause a mess and a hazard. So plates spend most of their life upside down. That's how we store them. That's how we incubate them. That's normally how we work with them, upside down. Now, when it comes to tubes, whether we have a plastic cap or a metal cap, these caps just push on and pull off. So they also do not seal. There are gaps to allow for gas exchange. So what that means is when you were working with tubes, there's only two places that a tube should be. You're either holding it in your hand or it's sitting in a test tube rack like we see here. You never lay a tube down because if it has any liquid in it, it's going to leak out all over the place. Also, when you're holding a tube, you can't turn it sideways, otherwise it will leak out all over the place. So again, these are things you would have to keep in mind as you're working in the lab. So, advantage of solid agar versus liquid broth. Any ideas of an advantage of using the agar versus broth? Anything? No ideas?
I think it's, yeah, okay. You said it.
So you can use solid agar to separate, right, bacteria, but who was that? I said that here. But Taylor said you can see, right, the bacteria on the surface of the agar. And that's kind of the main idea, is that if you had a broth culture like what we see here, it could just be one kind of organism. It could be 100 different organisms. There's no way to know in a broth culture. But on the surface of agar, you would see different characteristics. Not all bacteria form the same size colonies or the same color colonies or the same texture colonies. So there are characteristics you can see on the surface of a plate. So when you're separating mixed bacteria, you can see that you've actually separated them. You can see if there's more than one kind of organism present. On this plate here, there's only one kind of organism that's present, right? It's all the same kind of growth. But let's say you had a really tiny colony here, or maybe you had a bright yellow colony. That would tell you that there's a different organism on that plate. There's more than one of them there. So the advantage of the solid agar is that we can see the bacteria on the surface. We can use solid agar for isolation and separation of bacteria. So number two, an agar slant versus an agar deep. Well, we already talked about that, right? What's the advantage of slant?
More surface area.
Right, more surface area for growing our bacterial cultures. Agar deeps have specialized functions. We can stab them, let the bacteria grow, and observe a characteristic. Or if it's a large agar deep, we can melt it and pour that into an empty petri dish and make our own plate. But the slant has more surface area. Which container would provide the most surface area? Out of all the containers we see here, which one has the most surface area? Right, the agar plate, because we got a whole lot of surface area here, right? So lots of surface area. So number 4 is as simple as it sounds. What types of containers do we use? We use tubes and plates. So number 5, when it comes to liquids like broth, would you put broth in an agar plate or in a petri plate? Would you use liquids in a plate though? No, only in tubes, right? So when it comes to liquids, to broth, you only put broth in tubes, not plates. And what about agar? I mean, can you put agar in tubes? Sure, right? I mean, we see it here. But when it comes to petri plates, only ever agar in a plate. You can put agar in tubes, but when it comes to plates, you only ever put agar in a plate. No broth. So any questions on these questions? And this is being recorded. So the second page, in the past when we had a lab manual, we would ask students just to go through and find all these different things and talk about how they're used. So I'm just going to talk a little bit about that now. So these are some examples. We do sometimes use an empty tube, which is not sterile, but we sometimes use it. Here we have a tube that has soft agar in it. The screw cap tube also has a different type of soft agar in it, not very much in it. And here we have one of our large fat agar deep. So this is a much bigger tube with a lot of agar in it. And then here we have some tubes with different colored caps. So we do color code a lot of our media so that you can easily recognize and know what it is. So, and we do expect students to know what our color coding is. So our test tube was soft agar. So that was this one here. And so what you would do with this is you would actually stab it with bacteria. And then we would observe to see if the bacteria can move through the agar. So it's actually used to determine motility. But you have to use a softer, thinner agar so that bacteria can move through it. Regular agar is way too thick. It's way too dense. And so bacteria can't move through it. Test tubes with a screw cap. So we do have some screw cap tubes that we use. And so in this one here, which just has a little bit of agar in it, what we use this for is to mix a bacterial host like E. coli with a bacterial virus. And then we spread that mixture onto the surface of an agar plate. So that's an experiment we'll talk about later on, which was kind of fun to do. We were actually mixing a bacterial virus with bacteria, and we could observe infection happening. Okay, next. The test tubes with colored broth and also different colored tops. So these are these tubes here. So what these tubes are carbohydrate fermentation tubes. So we can use them to determine if bacteria have the ability to ferment different carbohydrates. And the red broth is actually a pH indicator because a common end product of fermentation is acid. So we will see a color change from red to yellow. And that's how we would know the bacteria can ferment. The type of carbohydrate is indicated by the color of the cap. Silver was mannitol, blue was glucose, red is lactose. And we'll talk more about that when we actually talk about this experiment later on. Now, what's hard to see in this image, unless you zoom it more, and I can't zoom it anymore, is that there are little tubes inside of the big tube, and they're upside down. We actually call those Durham fermentation tubes, and they are bubble caps. So if the bacteria also produce carbon dioxide gas when they ferment, you can trap the gas inside these little fermentation tubes, and that is an additional characteristic that we can use to identify unknown bacteria. We'll talk more about carbohydrate fermentation when we actually get to it in lab. We just wanted students to kind of get exposed to all these different things. And the empty tube with no cap. So we do sometimes use empty tubes with no caps. So it's an empty tube. That means it's not sterile. Why would we use an unsterile tube in microbiology? I mean, I just told you we sterilize all media so that only the bacteria we introduce will grow. So why would we want to use an empty tube? And I don't expect you to know that answer. But the reason why is in the future, when we're doing biochemical testing, you will inoculate broth tubes with bacteria, you incubate them, they grow, and the bacteria can produce different end products. And so you can pour some of that broth out into an empty tube, add some chemical reagents, and observe if that end product has been produced. Since the bacteria have already grown, they've already made their end product, it's okay to put them in a non-sterile tube because we're immediately adding chemicals just to look for the presence of an end product. So that's how and when we use those. Which container would you guess to be best for bacteria which cannot tolerate air? So, or more specifically, oxygen. What did we call those bacteria that don't like oxygen or don't need it? What are they called? Anaerobic bacteria, right? So if you're growing bacteria that don't like oxygen, which would be the best thing to use? A screw cap, right, Sarah? So the screw cap tube, you can, tighten, right? You can screw it down so that it's nice and tight so no air gets in. Remember I said that all these other caps have gaps in them, so they allow for gas exchange. But the screw cap, you can tighten that the cap down real tight so that no air gets in. But there are some disadvantages of a screw cap. Can you think of any disadvantages? What if an organism needs oxygen? Would a screw cap be a good thing to use? No. What if you have an organism that produces a lot of gas? Do you want to grow that in a sealed container? Yeah, it's not a good idea, right? So one of the problems is that there is no gas exchange with screw caps. So no air gets in, no air gets out. So if you have an organism that is a strict aerobe and really needs a lot of oxygen, it's not going to grow too well in a screw cap tube because it will quickly consume all the oxygen. And if you have another organism that makes a lot of gas, you really want that gas to be able to escape. So a screw cap tube is not the best, for certain organisms. Also, when it comes to handling a screw cap, one of the things we'll talk about and see an example of next class is that when you're working with cultures, you have to hold everything in your hands. So you hold the tube in one hand, and the other hand, you hold something like an inoculating loop. But you have to hold everything, and then you have to unscrew the cap with the hand that's holding the loop. So it's just harder to work with screw caps. So they're not as easy to work with, and they do not allow any gas exchange. So when it comes to transferring stuff, I just mentioned one of our tools, which is a loop, but we also use pipettes to transfer, broth cultures. We do frequently use the large pipettes like we see here, but in micro, we also use micro pipetters, which you guys are not going to be able to play with this semester, but we also use micro pipetters. And the main idea with pipettes is you always have to consider the amount that you're transferring and what would be the most accurate pipette to use to transfer that amount. We have a spreader here at the triangular shaped thing. This is used for spreading bacteria on the surface of agar plates. And this long metal rod here is an inoculating loop. It is the single most important instrument that you will use in a microbiology lab. You use loops for almost everything to transfer bacteria. Whether working from agar cultures or broth cultures, you use a loop. And you can't really see, it's hard to see, but there is a little loop on the end of this here, right, for grabbing the bacteria. But you use the loop a lot. And you sterilize the metal loop by putting it in the flame of the Bunsen burner, and that sterilizes it. and then you let it cool down, and then you can pick up bacteria. Now, I always like to make fun of students because they think that this loop is something really special, and so they hold it in very strange ways. But the loop is no different than a pencil. It's no different than a fork. It's no different than a paintbrush. If you're painting on a canvas, you hold the loop the exact same way. So it's not special in terms of how you hold it. Just hold it comfortably like you would hold a pencil or something like that. Next to it is an inoculating needle, also known as a straight wire, but it's not very straight because they get bent very easily. So you have to straighten it out before you use them. And there's two reasons why we use a straight wire. One is to stab an agar deep. So you use a nice, needle, a straight wire to stab the deep, so you get a nice, neat stab. You can also use this to pick colonies off the surface of an agar plate, because maybe you have a really, really small colony on the plate. It's much easier to pick it with this tiny end of the needle than it is to use the loop. Or maybe you have two colonies that are really close together and they're different things, so you don't want to mix them together. So it's easier to use the needle just to grab that one colony without touching the other one. And then we have swabs. Sometimes we do use swabs. You can use swabs to inoculate cultures, although that gets expensive because you're wasting swabs. Or you can use swabs to pick up a sample from the environment or from a patient, and then use that swab to inoculate something. So, and we'll talk about an experiment today where you guys would have used some swabs. So when it comes to the pipettes, again, the main idea was which pipette do you use for the most accurate transfer, or the most accurate volume? So we have these large fat pipettes and the skinny pipettes. What's the total volume you can transfer with the big fat pipette? So how much can you transfer with the big pipette A? Right, 10 milliliters, right? And with the little skinny pipette? 1 milliliter. All right, so there's a 10-fold difference between the two pipettes. So if you look at section X, the big pipette, what's that volume here? How much is that? It's 1 milliliter, right? So the same section on the little pipette, how much is that? It's 0.1 milliliters, right? And we said there's a 10-fold difference between these. And the section labeled Y here is how much? 0.5 milliliters, or half an ml. And this diagram is wrong. It should stop right here where I'm pointing. So what's the volume there from the 4 down to where I'm pointing? It's 0.05 milliliters, right? So 0.05, because again, there's 10 times difference between these, right? So if you were pipetting 0.1 milliliter, what's the most accurate pipette to use? If you're transferring 0.1, which pipette to use? All right, B, the little one, right? The 1 ml pipette, because that gives you the most accurate measurements. I mean, there are 0.1 milliliter lines on the big pipette, but that's really not going to be accurate. What if I asked you to transfer 7 milliliters? Which pipette would you use? A. Yeah, but can't you use B and just do it 7 times? But what's the problem with doing that? Get more work. Remember, you're getting paid by the hour, so no one cares. Right. So Taylor, it's not efficient, but it's also error, right? Because whenever you pipette, is it really going to be exactly, right, the amount that you think it is? No. So there's always going to be a little bit of error. So if we were to use this pipette and transfer 1 ml 7 times, each transfer has some error. And so you're compounding that error. So that's why just using the large pipette to transfer 7 mLs would be the most accurate. So once again, always consider the volume that you're transferring and what would be the most accurate pipette to use. So any questions? No? All good? I'm going to stop sharing this and look at the next one. So the environmental plate is an experiment you actually would have done on the first day of class. And the main idea here is that each table would have functioned as one large group, each table having 6 students. And then each student would have tested a different treatment. And so you would have inoculated TSA plates, and then we would have incubated them and then looked at those results actually the following week. And the reason for that is not everything that you would collect from the environment likes to grow at 37 degrees. So we would incubate the plates for 37 for a couple days, and then we take them out and leave them at room temperature for the rest of the time. So that allows many different kinds of microorganisms to grow. But each student would have gotten a TSA plate. Some of you would have used swabs. And then, of course, you label the plates. And remember, what side of the plate did I say you're supposed to label? Where's the writing supposed to be? Where's the writing supposed to be? On the bottom, right? So on the bottom of the plate. And so you would have labeled those with your name and the treatment that you did. So let's look at our 6 treatments. One of you would have rubbed your fingers on the surface of the agar plate. So I actually would have told you guys not to wash your hands at the start because we needed some dirty fingers to test. And then, of course, all the students who do that, they always want to wash their hands after they rub the surface of the agar plate. But the agar is sterile. Your fingers are dirty, but students want to wash their fingers after touching the sterile agar, right? Which is kind of funny. And then somebody else would have taken their plate and they would have opened it and just let it sit on the tabletop exposed for at least 30 minutes. Someone else would have taken a swab and pretended to brush their teeth and gums. And then they would have inoculated the plate. And, you know, for the afternoon class, that means you probably just ate and didn't have a chance to brush your teeth, right? And then someone else would have taken a swab and dipped it into a soil water mixture that we would have had for you and spread that on the surface of a plate. Someone else would have dipped a swab in a beaker of water from one of our fish tanks and spread that on the surface of an agar plate. And then the last treatment would be to test the desktop. And so again, normally on the first day, I would not let you disinfect the desktops just so we could see how dirty they are. But in the case of the desktop, you would take your sterile swab and dip it into sterile water first, and then you would swab the desktop. Why do we do it like that? It's not necessary for anything else we were doing, but why do we need to wet the swab with sterile water first before swabbing the desktop?
bacteria stick better onto it?
Yes, they would. So that's the whole idea, because the swab is dry and the desktop is dry. So you're not going to pick up as much stuff. So if you wet the swab first, then you can pick up a lot more stuff from the desktop. Now, after you use that sterile water, we can no longer guarantee it's sterile, right? Because, first day of lab, you guys are new, you don't really know what you're doing, you're not very careful, so we can't guarantee that that tube of sterile water is sterile anymore. So you would actually discard that after dipping your swab. And I know that sounds kind of wasteful, right? But that is what we need to do, because if someone else were to use that same tube, it may not be sterile anymore, so that would contaminate their results. So you would have discarded that tube. Now, I talked this morning about waste, so I'm going to give you the same speech. So all the plastic and metal caps are always washed, re-sterilized, and reused. The tubes, depends on what type of tube it is. Some of the screw cap tubes we also wash, re-sterilize, and reuse. But many of the tubes are actually just discarded. Although it's possible the glass may be able to be recycled, it's normally just discarded. And that does sound wasteful, but if you've ever been to a doctor's office or a clinic and maybe you needed a few stitches because you cut yourself or something, they have these cute little sterile packs which contain sterile scissors, sterile forceps, some gauze, and of course the sutures and the needle for stitching everything up. So you have these wonderful metal scissors and forceps. What do you think they do with them after they finish using them on you? I mean, do they put them someplace where they can be re-sterilized and reused, or do they just throw them in the trash? Right, they throw them in the trash, which is such a waste, but it costs more money to re-sterilize and repackage them. And it also poses a health risk because all that stuff is contaminated, so it has to be handled very carefully. So in fact, there is a lot of equipment that just gets thrown away after a single use because it's not cost effective, right, to try and clean it, re-sterilize it, repackage it. And it's also a safety issue. And it's the same kind of thing with the masks that health personnel wear. And you probably have a lot of those masks, the kind that's kind of like an accordion, kind of folded up a little bit. And you can buy them pretty cheaply now, like $1 a piece at the grocery store. But what the normal procedure was for healthcare workers is that they had to change those masks and throw them away after each visit. So they actually went through a lot of masks, on a daily basis. But early in the pandemic, when masks were at a shortage, right, didn't have enough masks, they were actually told to wear the same mask all day long, and in some cases to wash or re-sterilize those same masks. And so that is not proper procedure. And ordinarily, a healthcare worker, if they did not change their masks during the day and throw them away, they would have been fired because that violates the standard safety procedures. But because of the pandemic, they had to change how they did things because they just didn't have enough masks, right? So ordinarily, all those masks are disposable. You use them once and you throw them away, but they had to use them all day long. In some cases, they had to re-sterilize them and reuse them. So It does depend on the situation, but in general, there is a lot of waste because a lot of things are designed only to be used once because that's the most cost effective. It's also the safest thing to do. So again, safety procedures are important to follow, but unfortunately the situation required that we do things differently and not follow standard procedures. But you all have lots of masks now, right? You always wear them when you go outside somewhere, right? You better, Christiana. So, all right, so we've inoculated our plates. We're going to incubate them, and then we would examine them in the following week, and then we make some observations. So let's talk about those. So you would have written down for your individual plate what specimen you used, and then you look at the relative amount of growth. So you basically had a lot of growth, very little growth, or moderate growth. It was just a quality, type determination. Just look and sort of see and compare to everyone else's plates. And then you would make some descriptions of the growth on your plates because, as we already mentioned, not all bacteria look alike when they grow on the surface of agar. You can see different characteristics, different size colonies, different shape colonies, colonies with different colors and different textures. So you would make some observations and notice that you had more than one kind of microorganism growing on your plates. And then you would compare those plates and see which specimen had the most growth and which had the least growth. Now, when it comes to the least growth of all these treatments, we had fingers, air exposure, teeth and gums, soil, aquarium water, and the desktop. Which of those treatments do you think would have the least amount of growth? So which would you hypothesize to have the least amount of growth? Fingers, teeth and gums, soil, aquarium water, the air, the desktop. Which would you think would have the least? Okay, Christina says air, Rachel's air. And air typically has the least amount of growth, but it does still have growth. So there is stuff in the air. And so again, this is something you have to keep in mind as you're working in lab, is that you never open a tube or a plate until you're ready to inoculate it. Because the longer a tube or plate is open, the more likely contamination will fall in. So there is stuff in the air. And when it comes to the specimen with the most growth on it, it does vary from semester to semester. But any guesses as to what might have the most growth? Rachel says soil. Fingers. On your handouts, there is a picture of some example plates. Here we can see. And of course, they're labeled A, B, C, D for each treatment as it appears in the handout. But here we have air. This is desktop. And so one of the problems is that our lab coordinator, she is very compulsively clean. So she likes to disinfect the tabletops a lot. And so they're normally not as dirty as we would like them to do this experiment, but normally air has the fewest. And so when it comes to the most, this is fingers, air, teeth and gums, soil,